Hi everyone, my name is Leah Broger, and in this presentation, I'll be talking about the research I've been conducting this past year in the Merck lab, which focuses on creating a novel cancer vaccine delivery mechanism. And it involves these structures right here called liposomal spherical nucleic acids. But since that's a bit of a mouthful, we're going to call them SNAs for short. So to explain how these can be exploited for vaccine delivery, I will begin by elucidating the SNA structure. So spherical nucleic acids are a novel class of nucleic acids that exhibit completely different behavior from their linear analogs. And this is because the SNA architecture is defined by these dense, highly oriented nucleic acids, which are packed into this concentric morphology as shown by the middle figure. The core can be composed of different materials such as gold or other metals, but in my research specifically, I use liposomal SNA vesicles, meaning that the core is composed of phospholipid bilayers. And I do this because these are an especially attractive scaffold since they're completely biodegradable. In addition, liposomes are highly modular in their assembly, which allows for these constructs to be able to be tuned. And lastly, the hollow core of liposomal SNAs can encapsulate a variety of chemical cargo. And for the purposes of my experiments, I encapsulate them with tumor-associated antigens. And I will explain later on why this is significant. In addition, as you may have noticed, the constructs I create have these small protrusions, which are anchored effectively in the outside of the bilayer. And these are small fragments of DNA sequence that are designed to behave as adjuvants. Again, the significance of which I will make apparent later on. So given this tunable architecture, my research centered around improving the structure of these liposomal SNAs so that they could disintegrate properly and at due time when administered to the patients so that the vaccine could be released effectively. And this was centered around the premise that SNA constructs weren't remaining intact upon administration, effectively leading to a low co-delivery of the antigen and adjuvant to target cancer cells. Now, I've mentioned antigen and adjuvants quite a bit. So why are these relevant to cancer vaccination, you may ask? Well, in general, a cancer vaccine is comprised of two main components, which is where our adjuvant comes in. And this is a molecule that boosts the immune system and the response to recognize an antigen. And antigens are tumor-associated molecules that have inductible response on the immune system as well. So this is a simplified schematic overview that outlines the step of this process. But essentially what happens is that when these vaccine concoctions are administered, they are engulfed by dendritic cells. And this leads to the maturation of these dendritic cells, which is all that means is that the antigens are presented on the cell surface. This process is then iterated again, and these naive T cells become active T cells, which then go out, recognize, and kill cancer cells. And effectively, this innate machinery is what we're trying to upregulate and induce so that cancer cells can be killed effectively. There's a small caveat, however. And what happens when there are no known tumor-associated antigens? Well, that happens to be the case in triple-negative breast cancer. And the development of a therapeutic cancer vaccine for TNBC has been hindered because there are no tumor-associated antigens that have been identified. In fact, there's no estrogen, progesterone, or HER2 proteins present, hence the name triple negative. And also why I picked TNBC, because administering lysates with SNA is particularly favorable for this cancer, which doesn't have any antigens present. In addition, as an alternative to common vaccination strategies, tumor cells can be extracted from the patient, disrupted, and the protein can be isolated. And within this protein isolate, there are many lysates that can be behaving as tumor antigens, which is shown in the previous slide is necessary for an immune response to be induced. So ultimately, creating these tumor lysates have incredible benefits over normal vaccines because you have access to the total possible antigen pool. And you're also personalizing this to a patient's own tumor, since these tumor cells are extracted individually for each patient. So coalescing these two ideas together, if we package tumor lysates and SNAs, we could simplify and expedite common vaccination strategies by a massive amount. And again, this involves isolating tumor cells, breaking them down, encapsulating these into these vehicles that are then injected subcutaneously back into the patient. Now, to reiterate, my research is really focused on this latter part of the approach, which is the disintegration of the SNA when it's in the patient, so that the antigen and adjuvant can be effectively released. So how did I do this? Well, it was a two-pronged research investigation. Firstly, I looked at core fluidity and modulating that part. And basically, my hypothesis was that modulating the stability of the liposomal core of an SNA will impact the antigen delivery. And I did this by using 4 sets of liposomes, each composed of a single type of lipid, and these were prepared from lipids bearing different melting temperatures, but the same head group. Now, the reason why I use different melting temperatures is because, as you can imagine, different melting temperatures affect the fluidity of these liposomal cores and thus the way that they disintegrate in the body. Now, lipid chains with carbon-carbon double bonds, which are unsaturated, are more fluid than lipids that are saturated with hydrogens. And that's because on the molecular level, unsaturated double bonds make it harder for the lipids to pack together. And they put kinks in the hydrocarbon chain, as you can see. And so these molecules do not stack very well, and the intermolecular interactions are weakened. Second of all, I also looked at the DNA anchor. And specifically, I wanted to see whether using different DNA anchors could affect nanoparticle stability. And I did this by using anchors of different hydrophobicities. And hydrophobicity is a fancy word for the degree to which a surface repels water. And this, you can imagine, is important since it would affect the dissociation rate in our bodies, which are predominantly an aqueous physiological environment. So combining these two yielded the following results. Now, we looked at SNA stability by looking at hydrophobic anchors and different lipids of different melting temperatures. And we measured the rate of decay through FRET signaling. And this was essentially done by putting fluorophores, which are just bright lights, onto the anchors so that when these disassociated, the difference between each fluorophores could be measured and the dissociation rate would be calculated from that. AKA a loss of FRET signal meant greater dissociation. So using this data, this figure was created. And here we can see that the initial rate of decay was calculated for each construct, run in triplicate, and graphed in decreasing order of hydrophobicity and increasing melting temperatures. So DSPC had the highest melting temperature. And this is all as a function of initial rate of FRET signal decay. So what this graph is telling us in a nutshell is that SNAs are dissociating more slowly as higher melting temperature lipids are used and more hydrophobic anchors are used. And from these investigations, it was found that clearly SNA stability in the presence of serum is governed by both liposome membrane fluidity and anchor hydrophobicity. So given this, what are the following steps? Well, basically, we now know that liposome constituents and DNA anchor moieties affect SNA stability. So now we can test this in stability on vaccine efficacy by seeing whether each of these constructs, the ones that dissociate faster or dissociate more slowly, have a different propensity to stimulate DC maturation. Then we could also test the lysate cargo itself, which was the stuff that's encapsulated inside the SNA, and combine the most immunogenic construct from experiment 1 to the best lysate construct from experiment 2 to really create an optimum vaccine delivery vehicle. And ultimately, exploiting these parameters could spur the development of an incredibly effective therapeutic cancer vaccine for TNBC, which is exciting. And to that extent, I'd like to acknowledge the Merkin Lab and, of course, the undergraduate research grant for providing me with the funding to do this research. Thank you for listening to my presentation.